Students, today I bring for you a new chapter and that is excretory products and their eliminations. In this chapter, we are going to cover all the important topics of your NCERT and as well as your PDF. Okay, so here are the topics you can see. First is the human excretory system, second, urine formation, third, functions of tubules, fourth, mechanism of concentration of the filtrate, fifth, regulation of kidney function, sixth, micturition, seventh, role of organs in excretion, and disorders of the excretory system. We will cover NCERT line by line and as well as the PDF. Okay, so let's start. First of the topic, this one, excretory product and their eliminations. First of all, we are going to see what is excretion. You can see excretion is the elimination of metabolic waste material like ammonia, urea, uric acid, etc. from the body. Types of excretion: ammonotelism, ureotelism and uricotelism. First of all, we are going to see ammonotelism. Ammonotelism means it is the process of excretion of NH3, that is ammonia. Ammonotelic animals are aquatic invertebrates, aquatic insects, bony fishes and aquatic amphibians, etc. Because ammonia is highly toxic, so excretion needs excess of water and aquatic organisms get plenty of water. Ammonia is readily soluble in water and is excreted by diffusion through body surface or gill surface in fishes as ammonium ions. Kidneys do not play any significant role in its removal. Now coming to your NCERT, let's see what NCERT says. Animals accumulate ammonia, urea, uric acid, carbon dioxide, water and ions like sodium, calcium, potassium, phosphate, sulfate, etc. either by metabolic activities or by other means like excess ingestion. These substances should be removed from a body either totally or partially. In this chapter, we are going to cover actually how the elimination occur from our bodies. Okay. Ammonia, urea and uric acid are the major forms of nitrogenous waste excreted by the animals. Ammonia is the most toxic form. Remember the term, most toxic form, and requires large amount of water for its elimination. That's why aquatic animals, they excrete through ammonia, whereas uric acid, being less toxic, can be removed with a minimum less of water. The process of excreting ammonia is called ammonotelism. I told you bony fishes, aquatic amphibians, aquatic insects are ammonotelic in nature because ammonia is readily soluble, so it can easily soluble in water. And here remember, kidneys do not play any significant role in its removal. Okay, now coming to next. Types of excretion. Second term, ureotelism. It is the process of excretion of the urea. Ureotelic animals, that includes cartilaginous fishes, terrestrial and semi-aquatic amphibians, that are frogs and toads. Aquatic and semi-aquatic reptiles, alligators, turtles, mammals, etc. In liver, ammonia is converted into less toxic urea, so it needs only moderate quantity of water for excretion. Some amount of urea may be retained in the kidney matrix of some animals to maintain a desired osmolarity. Now, see this one, what it is said. You can see mammals, many terrestrial amphibians, marine fishes excrete urea, ureotelic, you can see, and here liver play an important role, and here the term osmolarity given. It is osmolarity given. Osmolarity means it is the measure of the total solute concentration within a specific volume of a solvent expressed in osmoles per liter or milliosmoles per liter. Okay. Now coming to reptiles, birds and land snails and some insects, they excrete nitrogenous waste as uric acid. in the form of pellet or paste with a minimum loss of water and are called uricotelic animals. Okay, there are three terms you can see: ammonotelic, ureotelic and uricotelic. Okay, now coming to the notes. Here the same terms are given, you can see. Now, excretory organs in animals. Here some excretory organs and seen in particular animals. First of all, protonephridia, flame cells. We all familiar with the term flame cell. Okay, it is seen in platyhelminthes, rotifers, some annelids and cephalopods. Nephridia seen in annelids, Malpighian tubules, insects. Antennal glands or green glands are found in crustaceans or prawns, etc. Kidney in higher animals. Now coming to human excretory system. Let's see what our NCERT said. Here, same terms are given. You can see protonephridia or frame cells are the excretory structure of platyhelminthes, flatworm example planaria, rotifers, some annelids, and the cephalopods. Amphiox. Protonephridia are primarily concerned with ionic and fluid volume regulation, that is osmoregulation. Nephridia are the tubular excretory structure of earthworm and other annelids. Malpighian tubules are the excretory structure of the most insect including cockroaches. Okay, same work. Okay, antennal glands or green glands perform the excretory function in crustaceans like prawns. Now coming to human excretory system. Here what your NCERT said. I hope you all remember in class 10th we all go through this term human excretory system, where you are familiar with some term and some new are there which we are covering this point in this chapter. Okay, in humans the excretory system consists of a pair of kidney. You can see there are a pair of kidneys are there, one pair of ureter, one pair of ureter. This is the first pair, first one, second one pair of ureter, and a urethra. This is the urethra. Okay, it is also labeled. Kidney are reddish brown, bean-shaped structure situated between the levels of last thoracic and third lumbar vertebra, close to the dorsal inner wall of the abdominal cavity. Last thoracic and third lumbar vertebra. The term comes from your vertebral column. In the next chapter, we are going through the vertebral column, where you can find this term again. And close to the dorsal inner wall of the abdominal cavity. Each kidney measures 10 to 12 cm in length, 5 to 7 cm in width and 2 to 3 cm in thickness, and an average weight is about 120 to 170 gram. Okay. Next, towards the center of the inner concave surface of the kidney is a notch called hilum. What is a hilum? Now this portion is called hilum. This one, this notch. Okay, this portion, it is called hilum, through which ureter, blood vessels and nerve enter. Inner to the hilum is a broad funnel-shaped space called renal pelvis. Renal pelvis. This is the term given. You can see renal pelvis with projections called calyces. It is not labeled here calyces. You can see these are the calyces. Okay. These are the calyces. This point. These are the calyces. Okay. Calyces. The outer layer of kidney is tough capsule. Inside the kidney, there are two zones, outer cortex and inner medulla. Cortex also labeled, medulla also labeled. Okay. Now, the medulla is divided into few conical masses, medullary pyramids projecting into the calyces. Let's see what the PDF shows. Here the same terms are given. You can see pair of kidney, pair of ureter, urinary bladder, urethra. Now, here you can see the diagram. It's the same as your NCERT, and the structures are given, names are given. Last thoracic and third lumbar vertebra, length also, average weight. Here you can see it is covered by 3-layered fibrous capsule. Okay, remember 3-layered fibrous capsule. Our kidney is covered and enclosed by a tough 3-layered fibrous capsule. Remember the term, questions are asked. Next, hilum or hillux, as I told you, this is the portion. The gapping portion is called hilum, okay? Through which blood vessels, nerve, lymphatic duct and ureters enter through the kidney. All the blood vessels are entered through the kidney through hilum. Here you can see this diagram, you can easily find out. Here the lumbar vertebra, these are the, okay, lumbar vertebra, okay, where the kidney is situated. Now, you can see on the concave side of the kidney, there is an opening, hilum or hilus, through which blood vessels, nerve, lymphatic duct and ureters enter to the kidney. Hilum leads to funnel-shaped cavity called renal pelvis. It is also labeled here, you can see, renal pelvis with projections called calyces. Here some major calyces, minor calyces, you can see. Okay. Next, a kidney has outer cortex and inner medulla. Medulla has a few conical projections, renal pyramids or medullary pyramids projecting into the calyces. Cortex extends in between the medullary pyramid as renal column and called columns of Bertini. Here you can see cortex extends in between the medullary pyramids as renal column that are called columns of Bertini. Renal columns are also called columns of Bertini. Okay. Let's see what your NCERT says. Here, columns of Bertini is also labeled in a bold one. Each kidney has nearly 1 million complex tubular structure called nephrons. You all familiar with the term nephron? How the structure of nephron? Which are the functional units. Each nephron has two parts. Each nephron has two parts. The glomerulus and renal tubule. Glomerulus is a tuft of capillaries formed by the afferent arteriole, a fine branch of renal artery. Renal artery and blood from the glomerulus is carried away by the afferent arteriole. Okay. The renal tubule begins with a double-walled cup-like structure called Bowman's capsule. This term also you familiar with, which includes the glomerulus. Glomerulus along with Bowman's capsule is called the Malpighian body or renal capsule. The tubule, renal capsule is also labeled here, Malpighian bodies. Next, the tubule continues further to form a highly coordinated proximal convoluted tubule. Okay, some further points are given, you can see here. Afferent arteriole, efferent arteriole, you can see blood comes inside the glomerulus through afferent arteriole and then from glomerulus it then goes out from the efferent arteriole. Okay, Bowman's capsule, and then here it is given the tubule continues further to form a highly coiled proximal convoluted tubule, PCT. In shortcut we can say PCT. Okay. A hairpin shaped Henle loop. Henle's loop is the next part of the tubule which has a descending and an ascending limb. You can see Henle loop. This is a loop of Henle, descending loop of Henle. This one descending one and ascending goes upside. Okay, it is two part. The ascending limb continued as another highly coiled tubular region called distal convoluted tubule, DCT. Here the DCT are distal convoluted tubule, this portion. Okay, this portion is called distal convoluted tubule. Next, the DCTs of many nephrons opens into a straight tube called collecting duct. This portion is called collecting duct. This one. Okay, collecting duct. Here there is another nephron is there. Okay, here another nephron is there. Okay, so many nephrons are connected to a single collecting duct. Next, many of which converge and opens into renal pelvis through medullary pyramid in the calyces. Now coming to the notes, that PDF you can easily find out, clear your doubts from this PDF and diagrams. Here, it is the renal artery. See the diagram. renal artery and how it goes inside the kidney. Nephrons, you can see structural functionality of kidney. Each nephron has two parts. One is glomerulus, second is renal tubule. It is a tuft of capillaries formed by afferent arteriole. It is called tuft of capillaries. Okay, this portion glomerulus is called tuft of capillaries. Means most all the capillaries are trapped here. You can say tuft of capillaries formed by afferent arteriole. Afferent arteriole. Okay, it is a fine branch of renal artery. Blood from the glomerulus carried away by the efferent arteriole. Then it goes out through the efferent arteriole. Okay. Next, it begins with a double-walled cup-like Bowman's capsule which encloses the glomerulus. Bowman's capsule labeled here. Glomerulus plus Bowman's capsule is called Malpighian body or renal corpuscle. Glomerulus plus Bowman's capsule. Both are called Malpighian bodies or renal corpuscle. Now, tubule control proximal convoluted tubule PCT. Here it is also marked Henle loop and distal convoluted tubule DCT. Henle loop is a hairpin shaped which is descending and ascending limbs. The DCT of many nephrons opens into collecting duct. Now, collecting duct extends from cortex to inner part of the medulla. Here you can easily find out. This portion is cortex. Okay. This portion is medulla. Okay. Here you can see collecting duct extends from cortex to inner part of medulla. The converge opens into renal pelvis. The converge opens into renal pelvis through medullary pyramids in the calyces. Malpighian body, PCT and DCT are situated in the renal cortex. Malpighian body means glomerulus and Bowman's capsule. PCT and DTC they are present in the cortex region, while the loop of Henle and collecting duct both are loop of Henle dips into medulla. Okay. Next, the efferent arteriole emerging from glomerulus forms a fine capillary network that is called peritubular capillaries around the renal tubule. Here you can see it is labeled peritubular capillaries. The efferent arteriole emerging from the glomerulus forms a fine capillary network that is called peritubular capillaries. Okay, around the renal tubule. A minute vessel of this network runs parallel to the Henle's loop, forming a U-shaped vasa recta. U-shaped vasa recta. Okay, it is called vasa recta. Next, types of nephron. Nephrons are two types. Cortical nephron, which are 85% found in most of the organisms, and juxtamedullary nephrons. They are found 15%. First of all, cortical nephron. In this, the Henle's loop is short and extends only very little into the medulla. Vasa recta is absent or highly reduced. Cortical nephrons की बात हो रही है. Okay, where the Henle loop is short, as you can see, loop of Henle is short one in cortical nephron and extends only very little into the medulla. Here the cortex portion from here to here, and here from medullary portion. Okay, cortical nephrons means the loop of Henle is short one. Okay, only slightly it is present in the medulla, and vasa recta is highly reduced, mostly absent. But the juxtamedullary nephrons, which are present in 15%, in this Henle's loop is long and runs deep into the medulla. Vasa recta is present. Okay, it is long and deep into the medulla. Here all portions are there, and in medullary portion you can see wholly the Henle's loop is present. Now, coming to structure of kidney, let's see your NCERT what it says. Same points, because all the important points are in the PDF one which I opened previously with proper diagram. Here the same term cortical nephrons are there, juxtamedullary nephrons. I hope you got this term cortical nephron, juxtamedullary nephron in the diagram particularly. Here you can see there is no diagram in your NCERT. Here peritubular capillaries, minute vessels of this neuron fall into envelope loop forming U-shaped vasa recta. It is absent or highly restricted in cortical nephrons. Okay. Now coming to urine formation. Before starting this topic, let's see the PDF. Here, structure of kidney at a glance, you can see renal pyramid, nephrons, cortex part, medullary portion, and the nephron portion. Afferent arteriole, glomerulus Bowman's capsule, efferent arteriole, and then PCT, peritubular capillaries, DCT, then collecting duct, and within this you can see loop of Henle, vasa recta. Okay. Now, urine formation and psychology of kidney. Process of urine formation. There are 3 steps including the formation of the urine. One is your glomerular filtration, second reabsorption, and third tubular secretion. First of all, glomerular filtration. Glomerular capillary blood pressure causes filtration of blood through 3 layers. One is your endothelium, glomerular blood vessels, epithelium of Bowman's capsule, and basement membrane between these 2 layers. You can see in this diagram. This diagram is important one. Here you can see. It is the diagram, and podocytes. We are going to through this podocyte term. Food process, filtration slit, lumen of glomerular capillary, Bowman's capsule. Here the epithelium, basement membrane which is present between the 2 layers, and the endothelium. Okay. So glomerular filtration occurs through these particular 3 membranes or 3 layers. First of all, epithelial cells or podocytes of the Bowman's capsule are arranged in an intricate manner, leaving some minute space called filtration slit or slit pores. Here they are present in like this. You can see podocytes are present like this one, okay, like this, which make it is called intricate manner and leaving some minute space. Here you can see some spaces are there, okay, and these spaces are called filtration slit, okay, filtration slits or slit pores. Here you can see epithelial cells or podocytes of the Bowman's capsule are arranged in an intricate manner. Same points are given. Diagram is changed. You can see. First of all, glomerular filtration. Almost all constituents of the blood plasma except the proteins pass onto the lumen of Bowman's capsule. Means all the things, all the constituents of the blood plasma except proteins passes through the Bowman's capsule. Okay, here goes inside this Bowman's capsule. About 1100 to 1200 ml of blood is filtered by kidneys per minute. In constitute 1/5 of the blood pumped out by each ventricle of the heart in a minute. Here, the amount of glomerular filtrate formed per minute is called GFR, glomerular filtration rate, GFR. That is the amount of glomerular filtrate, the things which are goes inside the, which are present in the glomerulus, that is called glomerular filtrate, formed per minute is called GFR. Normal GFR is equal to 125 ml per minute, that is 180 liters per day. Okay. 180 liter of glomerular filtrate is produced daily, but 99% of this reabsorbed by renal tubules. You can see 180 liters of filtrate are produced, but 99% are reabsorbed by the renal tubules, that is PCT, loop of Henle, and DCT. So normal volume of urine released is 1.5 liter. So remember, out of 180 liter, only 1.5 liter goes outside of our body, and the rest are stored, restored or reabsorbed, okay, from the filtrate. Glucose, amino acids and sodium etc. are reabsorbed actively and nitrogenous waste are absorbed passively, okay. Passive reabsorption of water occurs in the initial segment of the nephron. Here the same terms are given. See the diagram. In this diagram you can see cortex, outer medulla, inner medulla portions, and how the nutrients are absorbed in PCT, then in descending loop of Henle, and then in the segment of ascending limb and the thick segment of the ascending limb, and then DCT, and finally in collecting duct also you can see some NaCl, urea and H2O are also removed to maintain osmolarity. Remember the term osmolarity. Next, PCT reabsorbs most of the nutrient and 70 to 80% of electrolytes and water. Only PCT are reabsorbed. Simple cuboidal brush border epithelium of PCT increases the surface area of our reabsorption. So questions will come in PCT. We can find out simple cuboidal brush border epithelium, okay, which increases the surface area for reabsorption, so that the PCT can reabsorb 70 to 80% of electrolytes and water, okay. Now, loop of Henle maintain high osmolarity of medullary interstitial fluid. Descending limb is permeable to water but almost impermeable to electrolyte. This concentrates the filtrate. In ascending limbs, minimum reabsorption occur. It is impermeable to water but allows transport of the electrolyte. They work oppositely, okay. So the filtrate gets diluted. In DCT, conditional reabsorption of sodium and water takes place. Collecting duct extends from cortex to inner part of medulla. It reabsorbs large amount of water to concentrated urine. It also allows passage of small amount of urea into medullary interstitium to keep up the osmolarity. As I told you, some urea also goes out from the collecting duct to maintain osmolarity. Third is tubular secretion. It is the third step. Cells of PCT and DCT maintain ionic balance, means sodium potassium balance, and acid base balance, that is pH, of body fluid by selective secretion of H plus ion, K plus ion and NH3 into the filtrate and absorption of HCO3 from it. Collecting duct maintain pH and ionic balance of the blood by the secretion of H plus and K plus ions. Next, urine formation at a glance. You can see diagram is important. Follow the arrow and you can easily find out where water, solute, filtrate and reabsorption secretion occurs. Counter current system. Before starting this, let's see your NCERT. See the urine formation. Same important points are given. 3 steps. Okay, 3 main steps is filtration, reabsorption and secretion. Okay, they occur in different portions of the nephron. Filtration, where you can find out this term, 3 layer, as I told you, endothelium of the glomerular blood vessel, epithelium of Bowman's capsule and basement membrane found between the 2 layers. The epithelial cells of Bowman's capsule are called podocytes. Okay, and filtration slits and slit pores. As most of the things except proteins pass into the lumen of Bowman's capsule, that's why it is process called ultrafiltration. Okay, GFR also given. Amount of the filtrate found by kidney per minute is called GFR. Okay, healthy individual approximately 125 ml per minute, that is 180 liters per day. And you can see kidney have built-in mechanism, regulation of glomerular filtration rate, juxtaglomerular apparatus JGA. It is a special and sensitive region found by cellular modifications in the distal convoluted tubule and the afferent arteriole at the location of their contact. In GFR, in the particular nodes, in the last PDF, we are going to through the JGA, juxtaglomerular apparatus, okay, where the renin is helpful in the GFR, okay. Now, reabsorption, active and passive mechanism, and tubular cells secrete substances like H plus, K plus and ammonia into the filtrate. Function of tubules, where you can see PCT is lined by simple cuboidal brush border epithelium that increase the surface area for reabsorption. Remember, question will come, okay? Simple cuboidal brush border epithelium. All essential nutrients, that is 70 to 80% of electrolyte and water are reabsorbed. It also maintains the pH and ionic balance of the body. Hydrogen ions and ammonia filtrate by absorption of HCO3 from it. Henle loop. Here the reabsorption occurs less quantity in its ascending limb. However, it plays an important role in the maintaining the high osmolarity of medullary interstitial fluid. The descending limbs of loop of Henle permeable to water but almost impermeable to electrolyte. Remember, the descending loop of Henle is permeable to water but impermeable to electrolyte. This consents the filtrate as it moves down, and ascending limbs work oppositely. It is impermeable to water but it allows transport the electrolyte actively and passively. Okay. Now, DCT, here you can see Na plus and water takes place in this segment. It is also capable of reabsorption of HCO3 and selective secretion of hydrogen potassium ions and NH3 to maintain pH and sodium potassium balance in the blood. Next, same diagram, you can see cortex portion, medulla portion, and how the things collecting duct occur. Here it is given, large amount of water could be reabsorbed from the region to produce a concentrated urine. This segment allows passage of small amount of urea into the medullary interstitial to keep up the osmolarity. Play an important role in maintaining the pH and ionic balance in the blood by selective secretion of H plus and K plus ions. Okay. Now, coming to the PDF. Consider the filtrate. The flow of the filtrate in the two limbs of the Henle lobe and the flow of blood through the two limbs of the vasa recta are in opposite direction. Here we are covering the topic counter current system. Okay. See this. The flow of filtrate and in the two limbs of the Henle lobe and the flow of blood through the two limbs of the vasa recta are in opposite directions. Okay. That is in the counter current pattern. This is called counter current mechanism. This is called counter current mechanism. Due to counter current and proximity between the Henle loop and vasa recta, osmolarity increases from cortex to the inner medulla interstitium, that is 300 to 1200. This gradient is caused by NaCl and urea. NaCl is transported by ascending lip of Henle loop and that is exchanged with the descending lip of vasa recta. NaCl is returned to the interstitium by ascending limbs of vasa recta. Similarly, small amount of urea enters to the thin segment of the ascending limb of loop of Henle, which is transported back to the interstitium by collecting tubule. Thus, electrolyte and urea are retained in the interstitium and maintain a concentration gradient, that is interstitial gradient, in medullary interstitium. It enables easy passage of water from collecting tubules to concentrate the filtrate, that is urine. Now, finally, the DCT and collecting duct produce urine 4 times concentrated than the initial filtrate formed, that is 300 to 1200. Okay. You can see initially it is 300 but later 600, 900 and then finally 1200. Next, micturition. Before starting micturition comes to your NCERT. Mechanism of filtrate, same points are given. Okay, 300 to 1200, how the ascending collecting table. Counter current mechanism also given. Okay, yes, you can see same diagram is there. It is diagram representation of a nephron and vasorector showing counter current mechanisms. Now, human kidney can produce urine nearly 4 times concentrated than the initial filtrate formed. Okay. Next, regulation of kidney functions. The functioning of kidney is efficiently monitored and regulated by hormones feedback mechanism involving the hypothalamus, JGA, and certain extent, that is heart. Okay, before starting the regulation of kidney functions, we have to complete the PDF. Let's remember micturition. The release of the urine is called micturition. Release of urine. How it occurs? You can see. First step shows gradual filling of urinary bladder. Second, stretching of urinary bladder. Then here you can see there are some stretch receptors are there. Stretch receptors on its wall send impulse to the CNS, central nervous system. Okay. CNS passes on to motor message. CNS pass the message to motor. Then contraction of the smooth muscles of the bladder and simultaneous relaxation of the urethral sphincter. Here urethra and here are the sphincter muscle present here. Okay. Then micturition occurs. Then the release of urine. Next, the neural mechanism causing micturition is called micturition reflex. Okay. An adult human exceeds 1 to 1.5 liter of urine, that is 25 to 30 gram of urea per day. Urine is light yellow colored, watery fluid, slightly acidic, pH is 6.0 and has an characteristic odor. Various conditions affect the character of the urine. Urine analysis helps in clinical diagnosis of many metabolic disorders and malfunctioning of the kidney. Example, glycosuria, presence of glucose in urine, that is called glycosuria, and ketonuria, ketone bodies in urine indicate the diabetes mellitus. Here in the diagram you can see blue color, no glucose, green color, trace of glucose, that is the Benedict test of glucose. Okay. Orange, moderate amount of glucose, and brisk rate, that is large amount of glucose. Next, role of other organs. Let's see your NCERT. Here you can see, before starting the regulation of kidney functions, let's start the micturition, which is the topic we go through in the PDF. Micturition. You can see, micturition means release of urine from the body is called micturition. Okay. So as we all know, from nephrons it is then carried out to urinary bladder, then stored till a voluntary signal is given by the CNS. Okay. This signal is initiated by the stretching of urinary bladder and it gets filled with the urine. In response, the stretch receptors on the walls of the bladder and send signal to CNS. Stretch receptor present in the urinary bladder. The CNS pass on to motor message to initiate the contraction of the smooth muscles of the bladder, simultaneous relaxation of the urethral sphincter, causing the release of the urine. And here you can see the process of releasing urine is called micturition. The neural mechanism causing is called micturition reflex. And the points are given: 1 to 1.5 liter of urine per day, light yellow colored, watery fluid, slightly acidic, pH 6.0, and a characteristic odor because of chemicals present there. And then you can see on an average 25 to 30 gram of urea is excreted out per day. And there are some presence of glucose, glycosuria, ketone body, ketonuria, and urine indicative of diabetes mellitus. Role of other organs. Other than the kidney, lungs, liver and skin also help in elimination of the excretory waste. Our lungs remove large amount of CO2, we all know, approximately 200 ml per minute, and also significant quantities of water every day. Liver, the largest gland of our body, secrete bile containing substances like bilirubin, biliverdin, and cholesterol, degraded steroid hormones, vitamins, and drugs. Most of these substances ultimately pass out along the digestive waste. The sweat and the sebaceous glands, the skin can eliminate certain substances through their secretion. Sweat produced by the sweat glands is a watery fluid containing NaCl and small amount of urea, lactic acids, etc. So the primary function of sweat is to facilitate a cooling effect of the body. It also helps in the removal of some of the waste mentioned above. Sebaceous glands eliminate the certain substances like sterols, remember the term sterols, hydrocarbons, and waxes through sebum. This secretion provides a protective oily covering for the skin. Okay, and small amount of nitrogenous waste could be eliminated through saliva too. Now, see your NCERT. Lungs remove CO2, liver, bile, bilirubin, biliverdin, cholesterol, degraded steroid hormones, vitamins, and drugs. Skin, sweat, sebaceous glands. Here you can see water NaCl is small amount of urea, lactic acid, eliminate sterols, hydrocarbons, waxes through sebum. Saliva also eliminates small amount of nitrogenous waste. Regulation of kidney functions. See, it is done by hormonal feedback mechanism involving the hypothalamus, JGA, and heart. Changes in the blood volume, body fluid volume, and ionic concentration activate osmoreceptors in the body. Okay, all three: blood volume, body fluid volume, and ionic concentration. If there is a slight change, it activates the osmoreceptors. And then what they do? First of all, regulation by ADH. When body fluid level decreases, the osmoreceptors stimulate the hypothalamus to release antidiuretic hormone ADH or vasopressin. It stimulates water reabsorption from DCT and collecting duct. Thus, ADH prevents diuresis and increase body fluid volume. So, what it occurs? The ADH or vasopressin, it stimulates the water reabsorption from DCT and collecting duct. Okay. Thus, it prevents the diuresis and increase the body fluid volume. Okay. Dehydration, blood volume, salt ingestion, blood osmolarity, osmoreceptors in the hypothalamus, they goes to posterior pituitary, and ADH occur, which water retained by the kidney, and come to thirst, and we drink water. Negativity responses, blood volume increases, blood osmolarity decreases. Next, increase in the blood fluid volume, switch off the osmoreceptors. When we drink water, okay, that at that time fluid volume increases in our body, it switches off the osmoreceptors and suppresses the ADH release to complete the feedback. It is constrict the blood vessels, resulting in the increase of the BP, blood pressure. This increases the glomerular blood flow and GFR. Okay. Next, regulation by juxtaglomerular apparatus, JGA. This is special sensitive region that is called juxtaglomerular apparatus, JGA, formed by cellular modification of DCT and the afferent arteriole at the location of their contact. Here you can see juxtaglomerular apparatus. It is formed by the cellular modification of DCT and afferent arteriole where they contact. It is regulated by the GFR by renin angiotensin mechanism. A fall of glomerular blood flow or glomerular blood pressure, GFR, activates the JG cells to release renin. Then what occurs? Renin converts angiotensinogen into bloods to angiotensin 1 and then angiotensin 2. You can see renin converts Renin converts angiotensin to angiotensin 1 and then 2. It is a vasoconstrictor. Angiotensin 2 increases the glomerular blood pressure and thereby GFR. It also activates the adrenal cortex to release aldosterone. Okay, adrenal gland, they secrete aldosterone. Aldosterone causes reabsorption of the Na and water from distal part of the tubule. Okay, it increases the Na, sodium and water reabsorption in the DCT. Then this also leads to an increase in the blood pressure and GFR. Here you can see homeostasis, blood pressure volume, and stimulate the juxtaglomerular apparatus JGA respond to the low blood volume or blood pressure such as due to dehydration or loss of blood. Then again the cycle continues. Okay. Now regulation by ANF. ANF check on the renin-angiotensin mechanism. Okay. Increasing blood flow to the atria of heart causes the release of atrial natriuretic factor ANF. ANF causes vasodilatation, dilation of blood vessel, and thereby decrease the blood pressure. Next, disorders of the excretory system. Let's see your NCERT, what it says. Role of organs goes through this, and then here the term how it occurs, how the ADH hormone or vasopressin for the neurohypophysis, it facilitates the water reabsorption from later part of the tubule, thereby preventing diuresis. Okay, diuresis. And the increase in the body fluid switch of the osmoreceptors, and suppress the ADH. It also affects the kidney functions. Okay, and causes the blood pressure increase. Increased blood pressure can increase the glomerular blood flow and thereby GFR. JGA play complex regulatory role. Fall in GFR, a fall in glomerular blood flow or glomerular blood pressure, GFR activates the JG cells to release renin. It converts angiotensinogen in the blood to angiotensin 1. Angiotensinogen is a protein. Okay. When it comes in contact with renin, it converts to angiotensin 1 and then 2. It is a powerful vasoconstrictor. Okay, vessel constrictor, we can say simply. Increase the glomerular blood pressure and thereby GFR. It also activates the adrenal cortex to release aldosterone. It causes reabsorption of sodium and water from the distal part of tubule. It leads to increased blood pressure and the GFR because vessels are constricted, so blood pressure is increased and GFR also. This mechanism is called renin-angiotensin mechanism. An increase in the blood flow to the atria of the heart causes the release of atrial natriuretic factor. When the blood pressure increases in the atria, it releases the ANF, atrial natriuretic factor. It can cause vasodilatation, means the vessels are constricted, they again dilate it. Okay, dilation of the blood vessel, and thereby blood pressure decreases. ANF mechanism therefore acts as a check on renin-angiotensin mechanism. Okay, mechanism completed. Now coming to disorders of the excretory system. Let's see the PDF. First of all, uremia. It is the accumulation of urea in blood due to malfunction of the kidney. It may lead to kidney failure or renal failure. Okay, when the urea in the blood due to malfunction of the kidneys are not working properly, so the urea comes in the blood, it causes uremia. Okay, next, renal calculi. Stones or insoluble mass of crystallized salt, oxalates, etc. formed within the kidney. It is called renal calculi. Okay, next, glomerular nephritis. The inflammation of glomeruli is called glomerular nephritis. Here you can see this diagram. It is a healthy kidney, and this is a glomerular nephritis. How the area you can see glomerular portion is goes inside. Okay. Next, hemodialysis. It is a process of removal of urea in patients with uremia. The dialyzing unit, arterial kidney, contains a coiled cellophane tube surrounded by dialyzing fluid. You can see hemodialysis. Here, a dialyzing unit or arterial kidney, dialysis machine, it is dialyzer, okay, contains a coiled cellophane. In this, you can see there are coils are there, okay, these are called cellophane, surrounded by dialyzing fluid, and here inside this there is a dialyzing fluid is there, okay. Here it is the same composition of plasma except nitrogenous waste. Blood drained from a convenient artery is pumped into dialyzing unit after adding an anticoagulant like heparin. So blood drained from artery, okay. Here you can see blood goes inside, then pumped into dialyzing unit after adding anticoagulant like heparin. Anticoagulant, remove the blood from coagulation. That's why it is added heparin. The porous cellophane membrane of the tube allow the passage of molecules based on the concentration gradient. Next, as a nitrogenous waste are absent in dialyzing fluid, these substances freely move out, thereby clearing the blood through simple diffusion. The cleared blood is pumped back to the body through a vein after adding antiheparin to it. Then again antiheparin is added, then it goes inside the human body, okay, through vein, okay. Next, kidney transplantation. It is the ultimate method in the correction of acute renal failure. A functional kidney is taken from a donor. It is better to receive kidney from a close relative to minimize chances of rejection by immune system of host. The kidney transplantation. Here you can see this diagram. Donors are healthy kidney, recipient having diseased kidney. Both the kidneys are failed. So here you can see a kidney is transplanted. Okay. Now coming to your NCERT. What your NCERT says? Here, same. Accumulation of urea in blood is called uremia. Hemodialysis, artificial kidney, heparin, cellophane tube, all are, we can see this in the diagram. Antiheparin is added, okay. Kidney, renal failure, renal calculi, and glomerular nephritis. That is inflammation of glomeruli of the kidney. And with this, your chapter is completed. So watch this video, and from the next videos like this one, we will complete the NCERT portions, important terms, as well as the PDF. Okay. So thank you everyone for watching this video, and if you find this one helpful, then you can share it to your friends, and do not forget to subscribe my channel. Thank you. Lots of love from Jai Jagannath.